Are you tired of invasive procedures, imprecise neural control, and expensive and complex setups? There's a better way. With designer receptors exclusively activated by designer drugs, or DREADs for short, you can target specific neural populations with unprecedented temporal and spatial control without the need for invasive procedures. DREADs are genetically modified G protein coupled receptors, or GPCRs. GPCRs are the largest family of membrane receptors in the nervous system. They are essential signal transducers, where they turn extracellular signals into intracellular signaling cascades. GPCRs bind to intracellular G proteins that, when activated, trigger distinct signaling cascades. In neural contexts, this often leads to neurotransmitter release and reuptake, membrane excitability and action potential generation, changes in gene expression patterns, and synaptic plasticity, as well as structural changes to dendritic spines and axon terminals. What makes GPCRs so valuable for neuroscience research is their incredible diversity. GPCRs respond to a variety of neurotransmitters and neuromodulators, including dopamine, serotonin, glutamate, GABA, as well as hormones and environmental cues such as odorants and tastants. This is why GPCRs are implicated in almost every aspect of neural function, from basic sensory processing to complex cognition and memory, and why they're the target of approximately 30 to 40% of therapeutics. DREADs leverage this foundational GPCR architecture and engage these endogenous signaling pathways. They do so without disrupting native receptor function by modifying key binding domains. DREADs are activated by inert compounds such as clozapine N oxide, or CNO, which do not bind to any other receptor. Through viral vector transfections or transgenic mouse lines, these engineered receptors can be expressed in specific neural populations. This allows you to activate these specific neural populations through a simple injection of CNO.
Using DREADs is as easy as 1, 2, 3. First, you will need to design a construct that expresses the DREAD transgene, promoters to select the target cell population, and a fluorescent reporter molecule. Next, a viral vector will be inserted into the brain so that it can express the GPCR in the cell. There is an initial surgery needed for this intracranial delivery of the gene. Or alternatively, you could use a transgenic mouse line that already expresses the DREAD receptor that you need. 2 to 3 weeks post this surgery, the neurons will express these DREAD receptors that you've transfected. Then you will be able to reversibly activate or inhibit your target population shortly after the injection with your ligand with only a minor delay.
There are various different kinds of DREAD receptors available, such as those that bind to GS and GQ to increase neural excitability, as well as those that bind to GI to inhibit neural activity. There are also beta-arrestin pathway targeting DREADs to alter the intracellular pathways of the cell without G protein coupling. Using DREADs is a total walk in the park. Unlike with optogenetics, there's no need for implanted fiber optics cables.
No wires, no implants, no hassle.
Instead, CNO is used to switch the neuron on or off. This is much easier, preserves neural tissue, and is much less stressful for the animal. Doesn't he just look so much happier? The lack of implants means that you can also perform your experiments on multiple animals simultaneously. You're only limited by your experimental setup and the time it takes you to do your injections.
When using DREADs, the effects can last for hours compared to the millisecond to second range that you have when you use optogenetics. Another thing that's also possible when using DREADs is to use the same animal over multiple days in the same experiment. CNO has been repeatedly used in the same animals without any evidence of behavioral issues or hangover-like effects. Moreover, there has also been no signs of decaying efficiency. By training the animal with DREAD activation and testing it without activation, scientists are able to test different brain regions and see the effect on acquiring behavior versus performing behavior. With only those cells that express the vector being affected, you get maximal control when using DREADs. The targets of DREADs can be non-specific, neuron-specific, or cell-type specific. With dreads, you are also able to target deeper and larger brain regions by simply making multiple injections down that target region's axis. If we were to try that with optogenetics, it would be nearly impossible because the optic fiber arrays or cannulae would just create excessive damage. There are both excitatory and inhibitory receptor options when using dreads, so the sky's the limit.
But wait, there's more. Subdomain targeting is now available. You can now specifically target dendrites, axons, or spines. This is done by fusing the dread protein to a subdomain-specific targeting motif. For example, you can now specifically silence axon terminals by binding an inhibitory dread protein to an orexin motif. However, promoters can only do so much. If your construct becomes too large, it can negatively affect protein trafficking or function. Therefore, for greater selectivity, use transgenic animals. Various Cree lines are available in both mice and rats. The LOX-P sites are cleaved, and the DREAD transgene is flipped into its functional orientation. The DREAD gene is then transcribed with little to no leakage when Cree is not present. A wide variety of Cree-line mice and some rats currently exist in which the DREAD transgene is expressed in a particular subset of cells, including dopaminergic, GABAergic, and cholinergic.
We know you must be thinking, oh, that sounds amazing, but I don't really have the budget to just implement another technique right now in my research. We thought about that, and we are happy to let you know that DREAD is an incredibly cost-effective technique to implement. Aside from what is already available in a typical behavioral neuroscience lab and imaging facility, you'll only need a few items to get going with DREADs. You will need viral vectors or the transgenic mouse line, a stereotactic injection apparatus, and CNO. Furthermore, you don't even have to commit to using just one technique. DREADs can easily be used in conjunction with others, such as the Crelox-P system, optogenetics, or electrophysiological recordings. One big advantage of using DREADs is the fact that they use the cell's own GPCR endogenous signaling pathway. This means you'll be able to measure more physiologically relevant changes in the pattern of activity. Don't just take our word for it. Let's hear it from some real life researchers who have implemented DREADs into their work.
Hi, I'm Mark Ansorge, an associate professor of clinical neurobiology at Columbia University. In my lab, we focus on the origins of neuropsychiatric disorders such as anxiety and depression. Our research is mainly done using mouse models, and we use DREADs all the time. For example, we published a study a couple years ago that used DREADs expressed in the raphe nuclei to activate and inhibit serotonergic neurons. DREADs were the perfect technique to use for this, as they give a more accurate simulation of normal serotonin signaling. Previous studies utilized optogenetics, which results in fast, short-lived, and synchronous activation. This is a more artificial representation of this signaling, so we use DREADs as our technique of choice for a clearer view of these mood regulation pathways.
In addition to being a very useful investigative tool, DREADs are now being explored as potential treatments for neurological disorders. Let's hear from one of our expert researchers.
Hi, my name is Takafumi Minamimoto, and in my lab at the National Institute for Quantum Science and Technology in Tokyo, we look at epilepsy in non-human primate models. A few years ago, we published a study where we tested DREADs as a treatment for epilepsy. We used a viral vector to express the receptor HM4DI in neurons in the primary motor cortex in macaques. After inducing seizures in the monkeys, we activated the receptor, which has an inhibitory effect, and found that it was very effective at reducing the seizures. We also observed no significant side effects of this treatment in the monkeys. These results suggest that DREADs are a promising potential treatment for epilepsy in humans, with much further testing needed, of course. Instead of the current pharmaceutical treatments or surgery, DREADs could be a precise, effective, and reversible relief for patients suffering from seizures.
As you've heard, there are so many advantages when using DREADs, and the possibilities for applications are endless. However, there are some downsides.
DREADs suck! You guys suck! You guys are awful people! You hate DREADs! DREADs are terrible! Oh my god!
Firstly, the temporal precision is limited. And that is because dreads have a range from minutes to hours. And if you compare that to the range of optogenetics, who have a millisecond to second range, it's clear who would win. It is also important to note the potential for some dread variants to possess a level of basal activity, which would lead to a worsening of temporal specificity. Dreads also have a slow rate of washout, and that means it makes them impractical for slice physiology and bath application reversibility studies. Next is the issue of differences between species and brain regions. So, dreads differ when you use different animals and when you target different brain areas. Lastly, CNO has a potential of metabolizing back into clozapine, and that can trigger a variety of actions upon endogenous receptors of the cell. To prevent this issue from happening, we recommend you use newer ligands such as compound 21 or deschloroclozapine for better efficiency and potency.
Another potential hurdle is that inhibition or activation of a neural population is unlikely to be as robust as traditional methods, such as electrical stimulation to activate the population or microinfusion injections to inhibit them. This is because by nature of them being GPCRs, the effect of dreads is a naturalistic upweighting or downweighting of neural activity. This can be seen as a disadvantage if your goal is to completely silence all the neurons in your target population or make them go absolutely berserk. Similarly, it's important to note that not all cells within your target population will express the dread receptor. This is because even with the most effective viral vectors, 100% penetrance is not possible. For example, when using the common AAV8 synapsin viral vector, only about 2/3 of the neurons in your target population will express the dread. However, the non-dread expressing cells may still show changes in activity due to alterations to the circuit as a whole. As with any perturbation method, it can be unclear whether the behavioral changes associated with the neural activity are a result of cells expressing the dreads or the cells affected by the cells expressing the dreads. Our support team will be happy to help you get started with dreads in your lab. They can help you select the correct dread variant for your research question,
Design viral constructs with cell type specific promoters,
Establish reliable CNM dosing protocols.
and even help you develop validation assays to confirm the functional expression of the receptor. Dreads technology is continually evolving, with new variants and new ligands expanding the toolkit available to researchers. By incorporating this powerful technique into your neuroscience research, you unlock the powerful potential to exert precise control over neural circuits without the need for invasive procedures. Upgrade your neural control today. Call the number on the screen for a free consultation.
sorry,
Dread variant for
Shit. I'm sorry.
CZ, TCZ, DCZ.
family of membrane receptors.
Oh my god, electrophysiology.
I'm sorry.
The narrator is on a break right now. Can't come to the phone. We are performing a grand theft.
Test.
Hello.
Hi.
Oh.
Hi.
Hello.
A more accurate. Okay, wait.
Oh, fuck.
This is harder than it looks.
I'm not sure that's true.
Proteins. Absolutely berserk.
Don't drink and don't smoke.
Treatments for
That's a wrap. Mic drop.